Hello, and thank you all for watching. We measured the mating success of male Drosophila melanogaster developed at different developmental temperatures, as well as the interaction between developmental temperature and the performance or ambient temperature. Temperature, particularly in ectotherms, affects behavior and life history. What temperatures an organism experiences can differentially affect behavior, whether it is temperature experienced during development or temperature at the time of performance. Many alternative hypotheses have been proposed for the effect of temperature on behavioral outcomes, in this case applied to mating success. The beneficial acclimation hypothesis posits that an individual will experience the greatest mating success when their developmental temperature and performance temperature are matched. Alternatively, in colder is better, males raised at a colder developmental temperature will always outcompete males developed at a warmer temperature due to their larger size. Another hypothesis, warmer is better, was that males developed at a warmer temperature will outcompete males raised at a lower temperature due to having more energy available to dedicate to courtship. The optimal temperature hypothesis is similar to the previous two in that relative mating success is determined by developmental temperature and absolute mating success is impacted by ambient temperature, with the optimum at the intermediate temperature. The last hypothesis, developmental buffering, suggests that developmental temperature determines mating success, with ambient temperature having no effect. The availability of energy and general condition of an individual is greatly influenced by the environmental conditions in which the individual develops. The energetic cost of courtship may be expensive for individuals of poor condition, thus the quality of courtship should be indicative of the condition of the individual. The environmental conditions in which courtship takes place also impacts the ability to perform courtship, as well as female preference and tolerance window. When developmental and performance temperatures are varied, courtship ability may not only be reflective of the condition of the individual, but also the ability of the individual to perform the courtship in the environment. If the ability to perform courtship at ambient temperatures is positively correlated with developmental temperature or matched, then the beneficial acclimation hypothesis would be supported. One example of a signal in Drosophila melanogaster is courtship song, a signal significantly affected by temperature. Courtship song is produced by extending and vibrating the wing. This behavior may be energetically costly and therefore reflective of the condition of the individual performing it. While signals evolve to produce the highest quality signal at the lowest cost, developmental conditions may reduce energy availability below the level required to meet the cost. This is exemplified by individuals raised at a more permissive temperature performing pulses at a faster rate, or with a lower interpulse interval, or IPI. As shown in this experiment in which males were raised at 18 or 25 degrees Celsius and then their pulse song was measured. Song responds to temperature, as has been demonstrated before, with males singing faster, with lower IPI at higher temperatures. Males raised at lower temperatures sang consistently slower than males raised at the higher temperature. Because females may prefer males with faster pulse rates, absent of developmental effects, we hypothesize that mating success would be greater at higher performance temperatures, and females would always mate with males that developed at a higher temperature. In Drosophila, developmental temperature is negatively correlated with body size, and in Drosophila, females generally prefer larger males. Body size is also related to the energy available for courtship. However, one body size does not fit all. When body size differences are induced by food availability, larger males experience greater mating success. However, when male body size is influenced by temperature and tested at a single ambient temperature, the optimum temperature for life history traits, males do not experience greater mating success. As you can see, varying both developmental and performance temperature reveals a complicated relationship of antagonistic effects. We examined this relationship in Drosophila melanogaster. In the experiment, males were developed at 18 and 25 degrees Celsius. In each trial, males from both temperatures were placed with a female raised at a middle temperature, 21.5 degrees, for head-to-head mating competitions held at either 18, 21.5, or 25 degrees. Mating competitions were video recorded to determine who mated successfully and how much time each male spent courting. The wings of the males were measured as a proxy measurement for body size. The progeny of mated females were counted. As in previous research, males raised at a colder temperature were significantly larger than males raised at a warmer temperature. We did not find that the larger male in each pair experienced different mating success than the smaller male at any temperature. Thus, the previous result is confirmed that size, when induced by temperature, does not affect mate choice. We found that the most males mated at the intermediate temperature of 21.5 degrees. There was not a significant difference in mating success for either developmental group at each temperature. The 18-degree trials producing the least amount of males who mated is in line with our earlier hypothesis that more males would successfully mate at high temperatures, though the highest temperature did not produce the most males who mated. There was a significant difference between males who mated at each ambient temperature, irrespective of developmental temperature. As a proportion of trials, the intermediate temperature trials had more males that mated than either other temperature. Neither male group produced significantly different amount of offspring, nor did either group produce a significantly different proportion of males. Of our 5 main hypotheses, none match the outcomes of our experiments. This figure does not represent actual data, but the takeaway is conceptual. It shows the mismatch of the outcome to any of our hypotheses and sums up that more males mated at a higher temperature and between group mating success differences were not significant, though it does share some resemblance to the optimal temperature hypothesis. Separately, the proportion of time spent courting did not vary significantly between the two male groups. We found that males raised at either developmental temperature did not experience different mating success or differences in resulting progeny. Our research confirmed that when induced by temperature, larger males do not mate more often. Potentially, the optimal temperature for life history traits may not be the optimal temperature for mating success. The interaction between developmental performance and life history temperature effects is complex and requires testing at more intermediate temperatures to untangle these relationships. There will also be value in testing with females raised at different temperatures, as their developmental conditions may impact their preferences of mate choice.